Hello everyone and welcome to protein synthesis. Now if you haven't watched the DNA structure video, which is on the normal biology page, please watch that first. Again, this is one of the tricky topics in biology. So there's two parts of protein synthesis. There is transcription and then there is translation. Now some examples use those words and some don't. Okay, so if you haven't heard those words, don't worry, you just need the process. So we're going to be talking about RNA, DNA and transcription on this first video, and then probably translation and some questions on the next video. So here we go. So quick question, what is the function of DNA? Now, if you're asked that question, there's only one answer you can say, and that is the function of DNA is to code for proteins. Now, not all of DNA codes for proteins. There are some parts that don't, but the parts that do are genes. Okay, there's some other parts which we won't get into now, but the parts that code for a protein are called genes. And protein synthesis is all about making these proteins from the original double helix strand of DNA, which we can see there in the picture. So we need to talk quickly about RNA. So DNA is deoxyribonucleic acid. RNA is ribonucleic acid. It's similar to DNA, but there are a few differences. Okay. And we'll talk about the differences in a minute as to what they are. It's just another type of nucleic acid like DNA. We need to talk about messenger RNA in a second, or mRNA, as it's known in exams and everything. Okay, so let's talk about the differences. This is ribonucleic acid, and as you can see, up here it's got the same bases, A, G, and C, but it has a different one here. So it doesn't have a T, it has a U instead. That's the first difference between RNA and DNA. The second difference is, as you can see, it is a single strand. Okay, and it's a lot smaller. Now, the reason we need, beeping. The reason we need RNA is because DNA will not fit through the pores in the nucleus, okay, but RNA will. So RNA is taking a message from inside the nucleus across to the ribosomes where these proteins are going to be made. So three differences. It has a base U instead of T, it's a single strand, and it is smaller than DNA. Okay. So the first part of this, transcription, and in this bit we're going to be copying the DNA and making some messenger RNA to take that information from inside the nucleus to the ribosomes. Okay, so here is our DNA in the nucleus, and we need to copy it. Now the only way we can copy it is to unwind it and separate it, because in between these bases here, in between all of these bases, are hydrogen bonds. So we can't copy it until it's opened up. So the first thing that happens is the hydrogen bonds between those bases are broken, and it separates into different strands, two different strands. Okay. Now, in the nucleus are lots of free nucleotides, free bases, and they will attach to their complementary base. So normally in DNA, write it down here, A goes with T and C goes with G. Now this is still the case when they're attaching, okay, but as messenger RNA doesn't have a T, it will have a U instead, like here. So whereas that's a T in DNA, it's a U in messenger RNA. And gradually, these free nucleotides that are inside the nucleus, we actually call the cytoplasm inside the nucleus the nucleoplasm, but you can just say cytoplasm inside the nucleus. Because they're complementary, they will very quickly attach themselves to the DNA until they reach a stop code, which means stop attaching. Okay. Now what we've got now is this is our messenger RNA, and this is going to leave the nucleus through a nuclear pore, and it is going to travel to the ribosome, which is outside in the cytoplasm. So now it says here the enzyme RNA polymerase then catalyzes the formation of the strong chemical bonds. They took RNA polymerase out of the GCSE, so do not worry about that bit at the bottom. As long as you are understanding till this point, we are absolutely fine. Okay, so you don't need to know the name of this enzyme. However, what it does is joins all of these nucleotides together. Once they're all joined and chemically bonded together, there it goes, and we've got our messenger RNA all in one piece. It's then going to leave the nucleus. So here it goes off, moving away from this unraveled DNA, out of the nucleus and into the cytoplasm. Now these two strands of DNA are still complementary to each other. So these hydrogen bonds reform, and the DNA is as it was before we started. So it unraveled, we copied it, and then it's raveled up again. And then it will go back into its double helix structure. Right, so let's see what happens next. Ignore that, please. And we are now on to protein synthesis translation. Okay, so we're going to have a look at translation, and this is when that messenger RNA has left the nucleus and it is now in the cytoplasm. So how does that messenger RNA become a protein? Translation is the process where the messenger RNA is read and made into a protein. It occurs in the ribosomes, and this is what happens. Okay, so this is our messenger RNA leaving the nucleus. So this is where we finished on transcription. Out it goes. And it's now in the cytoplasm. And a ribosome, which I always think it looks a bit like the shape of a cauldron, attaches itself to the messenger RNA. Okay. Now, each of these letters are put into groups of 3, and each 3 is called a codon. And what happens is, that we can see all our codons, is transfer RNA molecules have what we call anticodons. So you can see there that we've got where are we? U. Now normally that would be TTT, but because this is messenger RNA, it's UUU, and the opposite to UUU is the AAA. Okay, so this bit here attaches itself, and this down here, let's see what happens with it. Okay, so here we go. These are all complementary, so that's our second codon attached. There's our third. There's our fourth. Now look at what's happening down the bottom here. All of these, are joining together. So we are making a line of amino acids. Now the codons go off, these green squares go off, and we are making a line of amino acids, and eventually it reaches that stop codon saying stop copying, and we have made a line of amino acids and join them together, and this is called a polypeptide chain. Now we are almost at a protein. The difference between a polypeptide chain and a protein is that we have to fold up a polypeptide chain and then it will become a protein. Okay, so chemically they're the same, it's just not folded yet, so it's not quite a protein.